We've looked at graded potentials, now let's look at action potentials. An action potential, or otherwise known as an impulse, is a sequence of rapidly occurring events that first decrease and then eventually reverse the membrane potential. This decrease in the membrane potential is referred to as depolarization, and then it is followed by a restoring of the resting membrane potential, and this is called repolarization. At this moment in time, that is probably pretty unclear, and we'll look at a picture that will illustrate all of this in just a second. Action potentials are messages that occur along axons and along muscle fibers. You'll remember that it's graded potentials that travel along the dendrites and the cell body of neurons. During an action potential, voltage-gated sodium and potassium channels open in sequence. You might remember that there are leakage channels inside of a neuron, but there's also what are called voltage-gated channels. These are channels that will open or close depending upon what the membrane potential is like, and we'll look at this in more detail in a little bit. Another principle dealing with action potentials, and we'll talk about this in more detail later on, is this. According to the all or none principle, if a stimulus reaches threshold, the action potential is always the same. We'll explain what threshold is in just a minute, but basically the idea is this. Unlike graded potentials that have different amplitudes or sizes depending upon the strength of the stimulus, action potentials are all the same. There are not different sizes of action potentials. So in other words, a strong stimulus which generates a graded potential that's strong will not cause a larger action potential. Action potentials are all the same size, and again this will make more sense later on as we look at a picture that describes this. So let's refresh our memory. Here we have a neuron, and we have the dendrites, the cell body, and the axon. You might remember from the laboratory portion of the course that there's a portion of the axon here at the beginning called the axon hillock, and we'll talk about that a little bit later on. Action potentials are generated starting at the axon hillock if the graded potentials that are arriving at that axon hillock have caused the neuron to depolarize to a certain level called the threshold level, which is approximately -55 millivolts in many neurons. So let's suppose we have a stimulus like, for instance, some neurotransmitters that have stimulated the neuron at these dendrites. A graded potential will travel along the surface of the membrane of the dendrites and then along the membrane of the cell body. When the graded potential reaches the axon hillock, if it has taken the inside of the cell up to -55 or the threshold level, then at that point an action potential will travel down the surface of the axon. So you remember that a graded potential is decreasing in size the further and further we get away from the source of the stimulus. If that graded potential has not died down too much and the membrane potential at the site of the axon hillock is at the threshold level, then automatically an action potential will travel down the axon. So here we have a picture of an action potential, and let's look at each of the phases. This is a very important slide, and if you remember these series of slides, then you really understand action potentials. So we're going to start off with a stimulus, and that stimulus really is the graded potential that's arriving at the axon hillock. So what we've done is we've stuck an electrode at the location of an axon hillock, and a graded potential is arriving at that axon hillock, and that's the stimulus that's arriving right now. We're measuring the charge of the neuron right at the site of the axon hillock with the electrodes that are stuck in the neuron. And we're noticing at this point, since the cell is at rest, it's at -70 millivolts. But when that graded potential arrives at the axon hillock, it will cause the inside of the cell to become less negative. And we'll explain what causes this a little bit later on. So here we are, and we're measuring the charge just inside the cell membrane at the axon hillock. And we're seeing that it's rising. And we're going to get it on up to the threshold level, or the -55. So at this point, we're going to go into the first phase of the action potential, and it's called the depolarizing phase. As we can see, the inside of the cell is becoming less and less negative. We're depolarizing. You'll remember that we mentioned that a neuron is polarized when it's at the resting membrane potential. In other words, there's a difference in the amount of charge on the inside versus the outside. And when it's at rest, the difference is negative 70 millivolts. But now, during this action potential, this message that's traveling at that site of the axon hillock, we can see that the inside of the cell is becoming less and less negative. And we'll explain what causes this in just a second. This phase, during which there's becoming less and less of a difference between the inside and the outside, is called the depolarizing phase. And we can see that at this point that the amount of charge on the inside and on the outside is equal. And then it's going to keep going so that the inside becomes positive and the outside is negative. This depolarizing phase is due to some voltage-gated sodium ion channels opening up. You'll remember that there is a greater concentration of sodium ions outside of a neuron compared to the inside. And so when these voltage-gated sodium ion channels open up, that will cause sodium ions to flow into the cell. Since sodium ions have a positive charge, then with them flowing toward the inside of the cell, the inside is becoming less and less negative, or in other words, more and more positive. And so the inside becomes less negative, and eventually it gets all the way up to being positive. The next phase of an action potential is called the repolarizing phase. It's shown in red here. During the repolarizing phase, potassium channels are opening up, and those sodium ion channels have closed up. So now voltage-gated potassium channels are opening. And you'll remember that there is a greater concentration of potassium ions inside of the cell compared to the outside. And since these voltage-gated potassium channels are now opening, the potassium that are inside are going to flow out. Since they're flowing out, and because they're positively charged, then that means there's a positive charge leaving the inside. In other words, at that point now, the inside is becoming less and less positive, or in other words, it's becoming more and more negative. And so this repolarizing phase is due to those channels opening up, which then allows the potassium ions to flow out. As we mentioned already, the sodium ion channels are closing during this phase. And so then the inside is reverting back to the negative state. The last phase that's found in many action potentials is called the after hyperpolarizing phase. During the after hyperpolarizing phase, those voltage-gated potassium channels are still open. Remember, potassium ions are flowing out, making the inside less and less positive, or in other words, more and more negative. These potassium channels are still open, and the inside of the membrane is becoming even more and more negative beyond the resting membrane potential. Those potassium ions are continuing to flow out, and the membrane potential, again, as we just stated, falls below the resting membrane potential. Eventually, those potassium ion channels will close up, and the cell will revert back to the resting membrane potential.